gonna make.
Our first student presentation tonight is by Matt Gennard. Over the previous year, Matt worked under the supervision of Dr. Karen Kellogg at Skidmore College and researched the effect of hydroponically grown watercress on nitrogen levels in water from local manure ponds. Matt is a junior and will be continuing his research into next year. Enjoy.
So my name is Matthew Gennard, and the title of my project is The Effect of Hydroponically Grown Watercress on Nitrogen Levels in Water from Local Manure Ponds. So an issue that had been seriously affecting Florida for well over a year was the red tide. The red tide is a seasonal event in which algal blooms appear normally between the months of August through December. But the most recent one proved to be one of the longest and deadliest, lasting from November of 2017 all the way into February of this year, killing nearly 600 sea turtles and about 200 manatees. So one of the issues that contributes to the severity of the red tide is eutrophication, which is when high levels of nutrients are carried in runoff into water ecosystems, and algal blooms are able to form because of this abundance of nutrients, and these algal blooms are normally detrimental to the ecosystem. So one of the most serious effects that these algal blooms cause is hypoxia. When the algal blooms are decaying, a lot of oxygen is lost in the process, and any other life in that ecosystem is normally unable to survive after these algal blooms have taken a toll on it. So this leads me to my next point. Manure ponds are man-made ponds that promote anaerobic conditions and contain a high amount of waste, so they're really high in nutrients as well. So they actually exhibit very similar conditions to eutrophic water sources, but they can also sometimes contribute to eutrophication should they overflow due to runoff. So if eutrophication contributes to the severity of issues such as the red tide and other eutrophic sources of water, and manure ponds can contribute to eutrophication, I wanted to look into a way that I could remediate the effects of eutrophication, both in sources of water that have already undergone this process and in sources of water such as manure ponds, which contribute to eutrophication. So I decided to come up with a design for a hydroponic raft, which would grow plants without the use of soil in an attempt to take up some of the excess nutrients in the ecosystem and hopefully prevent further eutrophication. I also wanted to try and use plants that could be economically beneficial to farmers, since manure ponds are found on farms. And I hope to test these ponds in manure pond water samples as well. The plant I decided to use was watercress because it already grows near water sources, so it's better suited to survive in conditions similar to being hydroponically grown, and it can also be sold. So for the first experiment I conducted, I made rafts out of styrofoam, and I placed 9 plants in each of the rafts. So there were 2 tubs that were set up that had manure pond water and had these plant rafts in them, and there was also a control group which didn't have any plants or rafts. It just had manure water in order to try and monitor any possible microbial activity that could also contribute to the results. So 9 liters of manure pond water solution, which contained 1 part manure water and 2 parts tap water, were put in each of the tubs. And these tubs I tried to keep them out of the rain and out of the sun because the plants are actually seriously affected by heat, and I wanted to try and weatherproof the setup as much as possible. Each plant was measured individually, just as a kind of baseline, see how they were surviving, and samples were taken every 10 days and frozen for further analysis. So these are the plant lengths from each of the tubs, and they're all over the place, but the most important thing to conclude from this is that each plant at some point experienced a period of growth, meaning that they are able to survive in conditions as harsh as a manure pond. So to analyze the samples that I took that I'd frozen, I first removed them from the freezer and let them thaw, and I filtered 40 milliliters of each of these samples to remove any solid waste that may be left over from the manure pond. They were filled with poly vials, which are small vials compatible with the machine I was using to analyze the water. And I put the samples within each vial. So the way that an ion chromatograph works, which is the machine that I use, is you put each of the vials into the machine and it goes through one by one, and it pumps what's called an eluent into each vial. So the machine analyzes at what time ions fall out of the solution due to the eluent, and it creates a graph based on each of these ions. So this isn't my data, this is just an example of what an ion chromatograph might produce. So the machine calculates the area underneath the curve, and using known values, it's possible to create an equation to relate the area underneath the curve to the parts per million of any ion in the solution. So these are the nitrate levels for the first experiment, the first tub that I had set up, and they were all over the place. There isn't really a reasonable explanation for why they didn't turn out as they should have expected. There was a heat wave at the time, so the plants weren't surviving as well as they could have. And the methods at this point in the experiment still needed to be revised. And the same can be said for the nitrite levels. They're a little more hopeful. They exhibited trends similar to what I would have expected. You can see there's an overall decrease in the first system, and in the second system, the rate of change was decreasing. So, given all the issues that this experiment had, I needed to come up with a new design. So I decided first I wouldn't be removing the plants just in case that had any effect on their ability to take up nutrients. And each system, rather than being conducted in a tub outdoors, would be conducted in its own system that I had designed in a greenhouse underneath grow lights to decrease any variability in the experiment. So this is just a picture of each system. It was constructed out of PVC pipe, and you can see plants were put in holes throughout. So with the new experiment, I had to change a couple of different things. Firstly, the solution for the manure pond water was significantly different because at the time that I collected this manure pond water, it was actually, there was a lot more waste in it, and that wasn't really, it wasn't representative of what manure pond water would be in the summer or in the spring when you would likely employ these rafts. So what I did was I filled each system with 8 gallons of the solution, which had 1 part manure water for 7 parts tap water. I removed 50 milliliters of water at the time that I collected each sample. The water was frozen immediately, similar to the last experiment. Samples were taken on days 15, 21, 28, and 35. and water analysis was completed using the same methods as before with the ion chromatograph. So the nitrate levels exhibited trends more similar to what I would have expected in the first experiment. You can see that there's this initial rise in nitrates and then it drops off. However, the control group shouldn't have been following these same trends, and I still need to address that. There was a risk for cross-contamination, and microbial activity could have played some part in that. That still needs to get looked into farther. And the nitrite levels as well were relatively inconclusive. So there were a number of failed attempts for this experiment. Watercress was constantly dying, and the heat played a huge part in that, and the harshness of the environment that it was being introduced to all played a role in the ability for the watercress to survive. So there were a lot of limitations as well. The relatively small presence of watercress may not have been able to affect the nitrogen levels within each system. And like I mentioned earlier, there was the possibility of cross-contamination, and also the mixture most likely wasn't homogeneous. So the amount of waste in one spot in the system may not have been the same as the amount of waste in another spot. So ultimately, I need to improve my methods. I'm planning on increasing the amount of biomass present in each solution. I'm going to ensure that it's entirely mixed, and I want to eliminate any possibility for cross-contamination. And I think with better methods and more experience, I could, well, hopefully I'd see results similar to that of what I'd expect. So I'd like to thank my mentor, Dr. Karen Kellogg, for allowing me to use her lab and helping me with the design process. My UHS classmates, Mrs. Barber and the Barber Rose Farm, for supplying the manure pond water, and my teacher, Mrs. Dixon. Are there any questions?
The manure ponds are made to control runoff? Is that the point of having them? That they're these man-made ponds?
So the question is, are the manure ponds designed to stop runoff? And they are. There are certain requirements that the manure ponds have to meet, such as the thickness of cement that's used to create them. But just with rainwater alone, sometimes there's enough for the manure ponds to overflow and escape into the environment around.
Okay, so my next question is, they're all cement-based?
Yep.
Okay,
I believe they all need to be cement-based.
Yeah, so that I would. Yeah, and then is any part of this process filtering that, or is there anything that could filter it so that nitrogen, whatever, could be reused in farming?
Well, the idea actually is that with the watercress taking up the nitrogen, you're already using the nitrogen there, so you don't need to worry about it escaping and affecting other water sources while also putting it to good use and something that's economically viable.
Right. Very interesting. Good job.
Thank you.
Kyla Mays will be our next presenter, who is in her junior year. Kyla Mays' mentor was Dr. Flemishing at SUNY Albany and created a project entitled The Photodegradation of Microplastics. Kyla Mays will continue this research into her senior year.
Hi, my name is Cal Mays, and my project is entitled The Photodegradation of Microplastics in Water. Alright. We all know plastics have a widespread negative effect on the environment. These range from the ingestion by marine animals like the Midway Island birds or sea turtles. Because of our use of single-use plastics and the improper removal of these plastics, ocean pollution like the Great Pacific Garbage Patch are floating around wreaking havoc. The Great Pacific Garbage Patch is estimated to contain around 150 million metric tons of plastic and is added to per year around 8 million metric tons of plastic. Another characteristic of plastic that makes it so dangerous is its biological persistency and its ability to absorb chemicals like bisphenol A, or endocrine disrupting chemicals. These chemicals harm marine life even more than simply the ingestion of plastics. In the past few years, and in the next couple, there will be efforts to remove these plastics from the environment. For example, the ocean cleanup device was designed to round up loose plastic bags and bottles that make up the Great Pacific Garbage Patch. While this device broke earlier in the year, it was one of the more efficient methods of removing large plastic. Despite civilian efforts like creating bracelets like 4 Oceans out of plastic from the environment, many large corporations have not cut down on plastic use and production. But on the bright side, the UK has recently banned single-use plastic bags, Q-tips, and plastic straws. In New York specifically, Governor Cuomo also promised to cut single-use plastic like styrofoam containers, which was also banned in New York City recently. Regarding bisphenol A, plastics known to contain this chemical were banned from manufacture. My project, however, focuses on microplastics. These are tiny plastic particles that range from 1 nanometer to 5 millimeters in diameter and have been known to be consumed by microorganisms like ghost shrimp. Because these plastics are consumed by our food, humans are now eating plastic. There are 2 types of microplastic, primary and secondary. Primary microplastics are found in commercial products like shampoo or toothpaste, and they enter the environment through the incomplete removal from wastewater treatment plants. Secondary microplastics are plastics that enter the environment as larger plastics like bottles or bags, but are more likely, but as they spend time under the sun, they degrade into smaller plastics. Where the ocean cleanup and other devices focus on the removal of large plastics from the environment, my project aims at the removal of these microplastics that these methods will leave behind. The goal of my experiment was to determine a method of degradation of microplastic spheres, ultimately shrinking their diameter using UV light and varying concentrations of chemicals, and to determine the efficiency of this method as a way to break down plastic. I started with 4 solutions. These solutions were composed of 25 milliliters of water, 10 or 50 microliters of a soap solution, which was made of an 8:1 ratio of water to soap. They also contained 50 microliters of hydrogen peroxide and 50 microliters of nitric acid. Each solution had a magnetic stirrer added to it, which was designed to keep the microplastics in suspension while exposed to UV light. However, there was only 1 stirrer in the lab that I used, so each experiment had to be done separately and was very time consuming. Solutions were put in a UV light machine for 1, 2, and 3 hours with samples taken out every hour, and the diameter was measured of the spheres. Each solution began with 5 milligrams of microplastic, and the soap solution was added to keep the microspheres in suspension. Whereas solutions 1 and 2 contained 10 microliters of the soap solution. This was because the soap was added directly to the solution along with all the other chemicals, whereas in 7 through 9 and 10 through 13 or 15, I think, or 12, 10 through 12, the soap was added directly to the microplastics to determine if there was a significant effect on the degradation and keeping the microplastics under the water. The results of the UV exposure showed the appearance of clumps and shreds of plastics along with the spheres in the water as demonstrated here. The first data point on my graph shows the average diameter of a microplastic sphere before UV exposure in millimeters. The first data point in each cluster, like for 1 hour, 2 hours, and 3 hours, is from solution 1, the second point from solution 2, etc. After 1 hour of exposure, the microspheres showed an increase in diameter likely contributed to the heat generated from the UV machine. After 2 and 3 hours, the microspheres tended to show a diameter relatively similar to that of the initial diameter of 0.231 millimeters. This demonstrated that 3 hours of exposure was not enough time to effectively reduce the diameter of the spheres. However, according to research by Rocha and Cobbs in 2014, plastics require a minimum of 6 hours exposure to UV light to show significant results of degradation. This requirement of 6 hours was not able to be met in the lab because of safety constraints and time constraints. After analysis of diameter, a gravimetric study was conducted to determine if there was a loss of mass of the microplastics after UV degradation. I began with 3 solutions, one containing just 25 milliliters of water as a control, and 2 containing 25 milliliters of water, 50 microliters of soap added to the microplastics, 50 milliliters of hydrogen peroxide, and 50 microliters of nitric acid, and all began with 5 milligrams of microplastics. However, again, due to time, these solutions had to be put under the UV machine at the same time, therefore only one had the magnetic stirrer and produced viable results. After degradation, the solutions were poured into glass filters in a vacuum, and these filters were put into an oven and a desiccator for 15 minutes each, 5 times, and the mass was collected each time and averaged. However, when the solutions were vacuumed and put under a microscope, similar clumps and shreds to the ones seen in the plastic shown from simply UV degradation were shown in the solutions, demonstrating that the measurements collected in the following graph were not entirely accurate. Before exposure, there were 5 milligrams of microplastics in each of the skewed and need to be repeated. So what could this mean? This means that my data does not support UV degradation as a viable means of breaking down microplastics. This could be because of time constraints in the lab, the inability to keep the microplastics in suspension, or perhaps unclean glass filters and the loss of microplastics in the vacuum. While UV degradation was not effective in this experiment, I would like to continue the study with more exposure time and redo the gravimetric study. I would also like to look into the effects of a Fenton's reagent, another oxidative reaction with an iron catalyst, or the bacterium Idaenella sachiensis, which was discovered in the Great Pacific Garbage Patch and is shown to eat plastic. I would like to acknowledge my former mentor, Dr. Flushig, and the graduate students in his lab that supervised my experiment. I'd also like to acknowledge my UHS teacher and my classmates, and Mackenzie Dixon for the ghost troop photo. These are my references. Thank you. Are there any questions?
What's a Fenton's reagent?
It's a reaction that is similar to the one that I did in my experiment, where you expose it to UV light, but instead of hydrogen peroxide and nitric acid, you would have an iron catalyst added. Mm-hmm.
So, when the exposure to UV light is making it small enough and drying enough to make it non-harmful to is that probably?
So the question was, what happens to the microplastics after you degrade them, basically?
Yeah.
So when you degrade the microplastics, they're already really, really small. When I had them in the lab, it was basically a powder I was using. So when you expose that to UV light, it makes them into even smaller clumps and shreds. Unfortunately, this does not make them any less dangerous because ghost shrimp, like Mackenzie Dixon saw, do eat microplastics and the shreds. So another thing I would like to look into is exposing these clumps and shreds that degraded from the microplastics to the bacterium that I mentioned, and perhaps that's a more feasible way of getting rid of the plastic.
If you were to use this on a larger scale, how would you be able to add all of these chemicals in the same proportion into larger bodies of water?
All right, so the question was how applicable basically is my study. So if this were to be applied to, say, an ocean system, it would have to be some sort of machine that just floats around, sort of, and perhaps the water would flow through the machine and the plastics would be decomposed there. That's my best guess. That's something I'd like to look into also. Any other questions? Thank you.
Our last student speaker tonight is Lucia DeKeel. Under the mentorship of Dr. Eric Yeager of Albany College of Pharmacy, Lucia evaluated the role of lipids in influenza replication. Next year, Lucia will pursue a degree in biology at St. Lawrence University.
Hi, my name is Lucia DeKeel, and my research is entitled Evaluating the Role of Lipids in Influenza Replication. So, influenza is a very contagious respiratory illness that affects. What makes it most infectious is that it can be contacted through contaminated surfaces. To many people, it's nothing more than a mild illness like the common cold, and you recover within a week or two. But the reality of it that about 12,000 to 50,000 people die from the influenza virus in the United States each year. A lot of these people are from the older generations and the younger generations who don't have a good immune system, and for many immunocompromised people. Each year the flu is constantly evolving through its replication process, which makes vaccines vaccines don't always work for every single strand of flu, which is why a lot of people end up getting the flu even if they get the vaccine for it. So like I said, a lot of people, when they get it, it's nothing more than the common cold, because a lot of the symptoms are the same. You have headaches, muscle and body aches, fatigue. The main one that stands out that makes it the flu is a fever, which is, as soon as you get a fever, a lot of people will go to seek medical attention for antivirals, but a lot of people don't get a fever, don't get treated because they assume they have a cold, and all the antivirals that are currently FDA approved all are needed within 24 hours of infection. So if you don't get those, you can't take the antiviral and it won't work, so other medications are used to lessen the severity of the symptoms, but that's about all you can do. And currently for prevention, it's just getting the vaccines, which hopefully work for the strands that have come out this season, wearing masks around highly infected areas, and washing your hands frequently and keeping your hands away from your face. So the life cycle of the influenza virus starts with the influenza virus binding to the host cell. It goes and encodes its RNA and hijacks essentially the host cell and uses it to transcription replicate its RNA, and then it packages itself back up and essentially leaves the cell, and it harms it in doing so, and this is how it buds and releases multiple viruses, which then infect more cells. When the flu virus is replicating in the host cell, it makes many mistakes, because unlike human cells, when they replicate, they don't have basically an autocorrect. So when DNA makes a mistake when it's replicating in human cells, they can fix it, but because influenza doesn't have that, they are subject to more antigenic drift and antigenic shift. This is just antigenic drift or slight variations and mutations to the flu virus, and antigenic shift is more harmful because that's when totally new strands are created. Sometimes these mutations to the flu virus help them, and sometimes they hurt them, and when they help the flu virus, they're more harmful for us. So why is this important? So the greatest example of this is the Spanish flu of 1918. It was one of the deadliest flu outbreaks ever. There was 40 to 100 million deaths worldwide, and a third of the population was infected. It lasted 2 years, and there was multiple waves of it that continued to infect the population. And it wasn't that it was such like a deadly strand, it was multiple strands mixed together with different subtype viruses from human and swine and avian-like viruses combined. So this led me to lipids. Lipids have been shown in previous research for the flu virus to leave the host cell, it needs to leave on a lipid raft, which led me and my mentor to choose lipids as a focus. And previous research has shown that viruses are dependent on lipids to leave the host cell and in their life cycle. So we figured that if you take lipids out of a cell, that the flu virus wouldn't be able to replicate as much, or wouldn't be able to leave the host cell at all, and then it can't infect the person as badly. So this led me to the hypothesis that influenza is dependent on the host GCS enzyme and the lipids it produces for its life cycle. So to test this hypothesis, I used 3 different techniques: a polymerase chain reaction, a Western blot, and a plaque assay. Each of these target a different part of the influenza replication. The PCR targets the transcription replication of the RNA, the Western blot targets protein synthesis, and the plaque assay targets the budding and release of the flu. So a polymerase chain reaction essentially copies an RNA sequence and exponentially amplifies and generates millions more copies of that RNA sequence. And for this, I used a wild-type HeLa cell and a GCS knockout cell, which means that it is lacking this enzyme. And when you take away this enzyme, the pathway to get this lipid produced, which the influenza virus needs to leave the cell, would be blocked. So the first step in a PCR is you purify your RNA. So we took RNA from infected and uninfected cells with flu of both the GCS knockout and the wild-type HeLa cells. And when you lyse them, you're taking out their nucleic acids, which then you will be analyzing. And then after this, you put it through a spectrophotometer to determine the concentration and the purity of the RNA, which then you add a master mix with buffers and primers and RT to isolate the RNA. And after this, you analyze it with, you analyze the data and graphs that the thermocycler produces. So this is a PCR amplification graph. So every time that a dye binds to the gene every time it is produced. So we are looking at every time that this specific enzyme was the enzyme was needed was produced is it fluoresces and increases the amount of brightness. So when you're looking at this, you're looking at which data point crosses the threshold first, and how quickly it reaches the threshold is how quickly this gene was produced and how much of it was produced. So I used the control gene and the gene of interest, which was SM1. So this is a graph I got out from the GCS knockout cells and the HeLa cells. So the wild type showed that when it was uninfected, the fold change in the gene expression of the SM1 was low, and that's the same with the GCS knockout uninfected, which was expected. That was my control. In infected, the fold change in the gene expression was relatively low, which was about expected. And then the GCS knockout HeLa cells, the fold change in the gene expression was very high, which was actually kind of opposite of what we expected. So from this, the next part of my research is to do a Western blot. A Western blot, you are separating proteins, you're staining them, and you're transferring them onto gel, and then through that you can visualize them. And the visualization is bigger proteins can't travel as far through the gel. So this was kind of a control for my project, just to make sure that all of the cells would show up and the virus that we infected it with would show up on our gel. This is actual data from my research. This was to see if the flu H antibody would work on our cells, because the lab I was working in was having a lot of problems to getting it to show up. So the last step of my research would be to use this specifically in the GCS knockout cells, which we were unable to finish. And the final step of this research would be to use a plaque assay. So through this, I established a protocol that we could use. So plaque assays are commonly used in when you infect things with a virus. When you infect something with a virus, you don't want it to be too potent that it kills the entire host population, but you don't want it to be not enough that it's unable to infect anything and you don't get results. So this, we diluted it, so each time you take a viral stock into a set amount of cells and things that they're in, and then each time you are taking the thing you just put into the next one, but you're sort of diluting it exponentially. And these are the results that we had, that 10 to the -1 dilution was too great and it killed everything off, and 10 to the -4 wasn't enough. So in the future, when we do use the PRA in the GCS knockout cells, you would use a 10 to the -1 dilution in this to determine if the cells are dependent on this lipid. So in conclusion, I learned several techniques that are involved in the study of the influenza virus. My study is not completely conclusive, but I helped determine a protocol for the lab I was working in for the plaque assay and the PCR and the Western blot, which will be helpful in ongoing studies in Dr. Yeager's lab, where this research will be continued. And like I said, the Western blot needs to be completed, and more tests of the PCR need to be done to make sure, because our results were unexpected, to make sure that is what we're actually getting. And I would like to thank my mentor, Dr. Eric Yeager, Dr. David Clark, Mrs. Dixon, my UHS science teacher, the Albany College of Pharmacy Summer Research Program, which I was a part of, my UHS classmates, and my family. Thank you. Are there any questions? And those are the references.
Our guest speaker will be Glenn Saunders, Senior Research Program Director of the Center of Automation Technologies and System at the Rensselaer Polytechnic Institute. Working at CATS for 13 years, he is the founder of the Upstate New York Laboratory Robotics Interest Group, a non-profit advocacy organization. He has his bachelor's in mechanical engineering and master's in engineering from Rensselaer, and is a licensed New York State professional engineer and has 1 patent. Glenn Saunders conducts research and develops projects in advanced manufacturing, robotics, and automation, with focus in concept development, prototype creation, manufacturing, and systems engineering, project management, and commercial deployment oversight. He also works on battery storage at the Battery Working Group of the ASME Thermal Energy Storage Task Force. He is also a pre-college education. He is also part of the pre-college education chair for the Hudson Mohawk section of ASME, and serves as a faculty advisor to the student section of the organization. Please extend a warm welcome to our guest speaker, Mr. Glenn Saunders.
All right, thank you very much. Thank you very much for inviting me tonight. I really like these science events They're really fun to me. This is really cool stuff. It's great when you have an opportunity to showcase science and technology and engineering and that sort of thing, because that's kind of what I like. So happy to be here. Thank you for the invitation. And you guys are doing some really cool stuff. This is really impressive. I'll try to keep things brief. I only have, I think, 324 slides, so I'll keep it short. So I think we have a pointer here, and we have a forward. Yes, forward. Okay. So I'm actually going to start out with a confession. I know this is a science symposium. Science is great, but I have a confession. I'm not a scientist. I'm an engineer. You probably wouldn't have guessed that because of, I'm so fashionably dressed, and I speak so eloquently, and I interact with people so well. So you wouldn't guess that I'm an engineer, but in fact, I really am. So my first question is, how do you tell an extroverted engineer? Well, if you're talking to an engineer and he's an introvert, he'll be looking at his shoes. If you're talking to an extroverted engineer, he'll be looking at your shoes. Okay, so I'm not going to take a lot of time. I'm going to pontificate just for a few minutes on a topic that's near and dear to my heart, and that's manufacturing. And then we'll get into some fun stuff. So this illustrates what I think is. Can you hear me? Okay. Okay. So this illustrates what I think is very common perception of what manufacturing is. And this is from a long time ago. Manufacturing is always thought of as it's dirty, it's dangerous, it's boring, it's all of those nasty things. And one of my points for tonight is this is an old, outdated perception of manufacturing. Manufacturing is not that. Modern manufacturing is more akin to what we're looking at here. Manufacturing is very, very technology oriented. Most of the factories that I go into are clean, spotless, air conditioned, excellent working environments, great job opportunities for anybody who's out there looking at manufacturing. In case you can't tell, my talk today is a little bit directed to teachers and parents as well as you guys, students. I'd like manufacturing to be thought of as a real valid career opportunity, because it really is. Manufacturing is changing, it's changing a lot. This is actually a Harley-Davidson plant. I visited a Harley-Davidson plant in Wisconsin, and at that Harley-Davidson plant, it looks just like this. This is not a doctored photo. And in that plant, they're so careful about the product that they're making. They're making jewelry in that plant. They're so careful about the products in that plant that when you go in, if you have a belt buckle like us old guys do, you actually have to put a little cover over your belt buckle so you don't rub up against their product and scratch it. So manufacturing. Manufacturing is, there's a perception that we don't make stuff anymore. Everything is made in China. That is actually not correct. We're doing an awful lot of manufacturing in this country. And yes, a lot of the manufacturing, that old school manufacturing that's in this photo is gone. But the advanced manufacturing that we're doing today is all over the place. And there's an awful lot of good jobs. In fact, there's a real shortage of skilled labor in manufacturing right now. The American Welding Society says that in the next 10 years, there'll be a shortage of 600,000 welders. If you go to machinists, again, huge shortage of people who are going into as machinists. Every factory that we go into, I've been in this, basically touring New York State, talking to manufacturers for about over 20 years now, and consistently we keep hearing that it's so hard to find people in these plants, and these are good jobs. And there are also some excellent apprenticeships right now. For example, at Hudson Valley Community College, you get into the apprenticeship program for machining, your entire education will be paid for, you'll have a guaranteed job when you graduate, and the average salary for people graduating from that program is in the $50,000 range. So, free school, guaranteed job when you get out, and a great career when you finish up. And again, in New York, you don't think of New York as this manufacturing hub. And again, I'm constantly surprised by every time we go out, every time we turn around, we find, oh, look, here's another manufacturer in New York making really cool stuff that we didn't even know existed. So, for example, we have manufacturers in the aerospace industry. You don't think of New York as an aerospace manufacturing hub, but in fact, there's an awful lot of aerospace manufacturing that's done right here, transportation, engine production. Even leather goods, menswear. I visited a plant in Rochester that's making men's suits in Rochester, New York, profitably. Space products, Moog out in Buffalo area, all kinds of food products, all kinds of health products. Manufacturing in New York is very robust. Okay, I'm done pontificating. Let's have a little bit of fun. I'm a robotics guy. I've been doing industrial robots for a long time here. These are some of the robots that we have in our labs. Most of the robots that we have are sort of industrial robots. Perhaps you're familiar with the Baxter robot here, which unfortunately the company that makes this is now Valiant. So we have now in our labs 4 obsolete robots. This is a Stably arm. This is a two-arm robot by Scala that we use for some space research. Believe it or not, NASA is actually considering sending robots to space to repair satellites. Satellites that were never intended to be repaired. They're up there right now. The batteries are dead. They have no fuel. They're ready to fall down. But it's more expensive to send up a new satellite than to have to go up there and fix the ones that are there. So we're working with NASA on how to grab a hold of the satellite, cut some things, and open up some other things, and refuel and replace the batteries and that sort of thing. This is a little robot made by Universal Robots in Denmark. This is one of those new generation collaborative robots designed to interact to work alongside people safely. Some more robots. This is a little project we had with the Albany Police Department SWAT team. They were looking for a low-cost robot to go in and help them with barricaded gunmen and hostage scenarios. That was used for a number of years in the police department. This guy right here is kind of a creepy social robot. There's, I think, 17 actuators in his face, so it doesn't quite get the facial expressions right, but enough, close enough that it's like really not exactly there, kind of creepy. We have some robots. This is a fuel cell powered thing that we did. This is one of my latest toys. We have a nice robot here for doing some robotic welding. I've mentioned welding a couple of times here. Welding is an area that we're starting to get into. We're finding there's an awful lot of labor shortages and an awful lot of need. One of our clients is actually Fort Miller, which is right down the street here. Robots, like these industrial robots like these, are not taking jobs. And I know that kind of sounds like I'm making it up, but the fact is that studies show that companies these days who are putting in robots are actually adding jobs. 20, 30 years ago, you could make an argument that robots were being put in to replace people because they were cheaper per hour. That is no longer the case. All the easy stuff has been done. The people who are putting in robots today, and manufacturers are doing so because they have to survive. These robots are not replacing people in manufacturing. They're helping the people who are there keep their jobs, and the owners of those companies keep those companies. So we really need the people like you who are interested in technology to help us get robots into production so that we can continue the manufacturing base that we have here. And manufacturing, okay, my last pontification. Manufacturing is an important aspect of our economy. It's official government standard statistics show that 10% of the economy is due to manufacturing. In fact, it's really quite a bit higher than that. It's more like 30% of the entire economy can be attributed to manufacturing. And the spillover effects are, government statistics say that for every dollar in manufacturing, there's another 40 cents in ancillary economic impact that comes from it. Other studies have shown that economic impact is more like $3 for every dollar of manufacturing impact. Okay, last, several students here are talking about either microplastics or harmful algal blooms. And this ties in well with the Jefferson Project, which is a project at RPI in collaboration with RPI, with the Fund for Lake George and IBM to to study Lake George and help protect Lake George. The program starts by understanding how the lake system works. So it's covered with a bunch of sensors. So we've got weather stations up and down the lake, up in Minnehaha. We've got other sensors inside the lake to measure what's going on to the water inside the lake. And funny, one thing that I found out when talking to the IBM researchers is that the water inside of Lake George actually flows north and flows south slightly during different times of the day, which who would have thought? Anyway, and we've got stream stations because all the tributaries are also outfitted with instruments to see what's going on in the streams. So, again, as I said, I'm an engineer, but the scientists that we're working with want to know what's going on inside the lake. So they have a whole bunch of sensors they want to deploy on this thing called the SON down into the water so they can understand what's happening. And they said, well, you know, we could buy this thing and it kind of works, but doesn't really do what we want to do. Can you build this one? We said, absolutely, we'd love to, because that's what we like doing. So we built 3 of these guys right here, which is basically a floating platform where you can drop this on and a weather station on top. And of course, every time we talk to the guy at IBM, they're like, well, you know what, we'd really like to measure the surface temperature of the water. Can you figure this out? Can you add a rate to this and do the surface temperature, measure the surface temperature? And of course, we say, we'd love to, as long as we have at least one field retrofit, which requires us to go up to Lake George on a nice, warm, sunny day in July. And we managed to somehow squeeze in a service call on Lake George at least a few times a year. But what was fun about this project was not only because we're helping to study Lake George and help keep Lake George clean, is that we got to build stuff. So we got a bunch of undergrads, we built the prototype in the lab, and go out in the lake and we try it out. It kind of works good enough, so we go to the CAD and we start building stuff, and go to the machine shop and start building stuff, get a bunch of students, and we keep building stuff. Whoops, I turned it off. How did that happen? Did I turn it off or did you turn it off?
It's you.
It was you. Ah!
No, it's
see,
you.
Okay.
It's on the clicker.
It's on the clicker?
Yeah.
Here we go. Okay. So, yeah, we build stuff and we put properly some cabinets and work out through the winter and build some more stuff, start running some code, things start to take shape a little bit, got some mats and stuff, put on some solar panels, do some more machining. They have these nice big solar panels so we can run all summer long without worrying about it. Finally, ready to go, we put it on a trailer, bring it up to the lake, now the fun begins. Now, we get to go on a shakedown cruise. So we're up on Lake George, we got a bunch of students, and it's time to tow this out to where we're going to do some testing and find out that things kind of work not exactly where we want, but good enough. And we're trying to brainstorm and took a bunch of data and found out that yes, the thing actually does work pretty well. And so we ended up building all three of these systems. We have, this is the 3rd year now, a whole bunch of, I think it was about 30 undergraduate students helped build this. They've been operating on Lake George and also at Skaneateles Lake last year, where on Skaneateles Lake there's a problem with harmful algal blooms, and we helped better understand one of the mechanisms for harmful algal blooms that we think has really never been discovered before, which that discovery would only have been possible with all the additional instrumentation that we have on here, which includes all the stuff we're looking at here, but underneath an acoustic current Doppler and an acoustic Doppler current profile, we can measure what's going on with all the microorganisms and so forth in the water column itself. And we're talking to, and this is incorrect, it's not Rainy Lake, it's Chautauqua Lake out near Johnstown, where we'll probably be deploying one of the units out there to help them work on their harmful blooms that they have in there. And with that, I am going to stop so we can all go home. Does anybody have any questions? Excellent. Thank you, guys. The parents and grandparents, you should be very proud of the students in this school. Thank you.
Excellent. So I just want to, before I take a moment to recognize our university and high school science students, I just want to take a moment and say some thank yous. I want to say thank you to Dr. Baker, who served on our SRC committee this fall, and also Mr. Cronin for serving on that committee, and the science teachers at school here, Ms. Whitman, Mr. Manera, Ms. Bain, I think Mr. Kelly was there for a period. It takes a tremendous amount of people to approve these projects, and when I send an email out in the fall, generally everybody just says they'll be there, which is fantastic. I want to thank Mr. Fish and Mr. Neese for allowing us to have this course and all these students and help support these students and all the things that they do, because they are some really incredible students. And that brings me to you parents. Thank you so much. I know there's a lot of work involved. Some of you have to do strange things like digging zebra mussels out of lakes in the middle of the fall and winter, to buying things that you never thought you'd have to buy, and building projects with your students. Without your support, I know that this program wouldn't be possible. And it goes without saying, for our science research students, especially since they start in their sophomore year, the driving that you do to some of these locations, usually there's not a college right around the corner. We're very lucky to have worked with Skidmore, but many of our colleges, like Albany College of Pharmacy, are very far away, and so your support is really, really helpful, so we thank you for that. I'd like to thank Mr. Glenn Saunders for his presentation tonight. I think you're going to go out of here as the favorite symposium speaker, because everyone understood you for the first time. I think we've had a symposium speaker that everybody understood, and you just have a great disposition with the audience, so thank you so much for coming. I want to say thank you to Courtney Barber, wherever she is. It's totally black, which is fine. For all of your help and support, she takes over the 7th grade class for the second half of the year. Next year she'll be doing the entire year. That happens every now and then. She is kind of like she comes up and says, what do you need? And she just makes it happen, makes reservations, etc. So thank you very much. I want to say thank you to Warren, who's back there doing our technology for us, because I have no idea how to run the lights and sound. So thank you so much for stopping everything in your schedule and being here tonight. The transportation group, who has brought us to RPI and JSHS, without our bus drivers, and if you know of anybody who wants to be a bus driver, we could definitely use some more. Transportation department is really critical to this program running, and so we are very thankful for them. And last but certainly not least, the people that make this room shine and clean, and the facilities around here wonderful, and are pulling together tables and pulling together, finding skirts and everything that I need for this evening, and we'll make sure that everything is tip-top shape for tomorrow morning when the students come back. The custodian staff here is fantastic. I am extremely thankful for them on a daily basis. They deal with our projects, which aren't always clean, and science is messy, and they're very patient with us and work with us and are really here for the students. So if you get a chance to see any of them tonight, please thank them. They do a fantastic job. So hopefully, I'm sure I've forgotten a million people. I apologize if I have, but you know, it really takes a whole community to run a program like this, especially since we have students at a very young age in 7th grade doing research all the way up to their senior year. There's a lot of really good research, and there's a lot of good scientists and engineers out in this audience, and so my hope is that we have more and more people going and students going into these areas, because it is an area that is a huge deficit, especially for New York State. We need more young, bright people going into science and engineering, mathematics, etc. And we're continually trying to draw from other areas, because our bright students aren't stepping up. And so I'm hopeful that some of these things that your students were involved with will help inspire them to do this in the future. And you're probably going to get, if you do science and engineering in the future, especially science, grad school will probably be paid for you, so that's something to consider. So I'm going to just recognize the high school students really quick and what they've been working with, what they've been doing. You've already heard from our 3 juniors and seniors, but the first student I'd like to recognize tonight did a great Explorer Vision. The first 2 students worked together on an Explorer Vision project this year. It was a huge undertaking where they had to develop a technology that they thought could be used, and they looked into Stirling engines and all kinds of interesting ways of being more conservative with energy and producing more energy. So our first student is looking at magnetic bearings and what their effect will be in a system, and that's Will Hamilton. So come on up, Will. So Will's actually, we're going to meet with an engineer tomorrow, so wish him luck if you see him on your way out as he meets with someone tomorrow, hopefully do some research with. Our second sophomore who worked with Will in the Explorer Vision project, who is really interested in looking at catalysts and energy reactions in fuel cells, is, see, I finally got it right, David. It only took, what, how many months? 3 months? David Gabriel. Our next student that we'd like to recognize tonight is a junior who worked with those hydroponic rafts and will be hopefully building and working in greenhouses this summer. That's Matt Gennard. No, you're good. Our second junior, who will be looking at saving us from all the plastics in the world, Callum Mays. And finally, I'd just like to recognize our senior tonight. She is our only senior. She has, oh my gosh, I'm trying to remember. She won a $20,000 scholarship last year at RPI to Albany College of Pharmacy. She's won other numerous awards for her research, and actually her previous year research, just in case you were wondering, in case you parents were thinking that you had like the most worst project ever, you could just spend some time talking to the DeKeels, because Lucia was interested in looking at meningeal brainworm, and the best way to do that is to harvest deer feces and analyze it. So she spent her entire summer harvesting and collecting deer feces and evaluating it under the microscope. So if you thought you had it bad, you talked to the DeKeels. So Lucia's gonna go into science. I know she's gonna do fantastic things, and I'm sad because she's my very last DeKeel. I don't know what I'm gonna do now. So they're all gonna have to move back to the area and have children, so I have more DeKeels. So congratulations to senior Lucia DeKeel. So thank you so much for this evening's Science Symposium. I am pleased to announce this is the very first time I've ended not only on time but early. Just a reminder to all of the students to please take your posters home tonight. Not the poster boards, but the posters can go home. And thank you so much for all your support. It really is so meaningful. This is a fantastic way to celebrate our students and all the hard work they've done this year. So thank you so much and have a great evening.